Hi, everyone. Welcome to our seminar series on the cerebellum. Today, we're really lucky to have Matty Joshua present for us his work on the way the nervous system encodes reward and transmits that from the basal ganglia and other structures to the cerebellum. Matty finished his work with Steve Lisberger, where he described these very novel findings regarding how reward arrives to the cerebellum via climbing fibers, and he's going to tell us about how that information is encoded both in the basal ganglia and the cerebellum today. So, Matty, thank you so much for joining us today.
Okay, thank you for Leza for inviting me. It's a great honor. As I said, my name is Matti. I'm a researcher and Edmund and Lily Stafford Center of Brain Sciences in the Hebrew University. You can see here our nice building, all covered by these pyramidal neurons. I think they should be actually Purkinje cells. It will be much nicer, but maybe next time. I'll talk today about our work in our lab in which we study reward and movement signals in the basal ganglia and cerebellum. So feel free to stop at any time, ask questions. Okay. So there are two major subcortical networks in the motor system, the cerebellum and the basal ganglia. There are many similarities, structural similarities between the two. So, for example, both are embedded in cortex computation, but both also have access to movement directly through the brainstem. But these two different systems are thought to have different roles in the control of movement. So classically, the cerebellum was thought as a structure which is important for optimizing the movement, calculating how to perform a movement, the kind of learning that is thought to be implemented in the cerebellum is motor adaptation, in which and the kind of learning signals are motor or sensory motor errors, and of course there's a nice correlate of the climbing fibers that are thought to transmit this error. The basal ganglia seems to have a different role in controlling behavior. It's thought to be related more to which action to select, the kind of algorithms that are thought to be implemented in the basal ganglia are reinforcement learning, and the kind of learning from external rewards, and the kind of error that is thought to be coded in the basal ganglia is the reward prediction error. And of course, there's the nice correlate of the dopaminergic neurons. However, our research and many others have recently challenged this view. And in this talk, I want to show our recent studies in which we studied reward in the cerebellum and compared between the basal ganglia and the cerebellum in terms of coding of reward and movement. Finally, in the last part, if I'll have time, I want to talk about a paradigm in which we studied how reward impacts motor learning. So let's start. So the system we mostly study is the smooth pursuit eye movement systems of the monkeys. This is a system that is developed in primates and humans in order to track moving targets. And here you can see the basic setup in our lab. A monkey is sitting in front of a screen, and in front of them they see targets, and these targets move continuously, and monkey is required to follow them. So the X here represents the eye, the square represents the target, and what you'll see is the target will start to move, and you will see the monkey tracking it. So this is how it goes. So initially, monkey is fixating the target moves, and the monkey follows it very accurately. We can plot this behavior and show it as a function of time. So the x-axis down here is the time, the y-axis is the velocity, and you can see that after a short latency of about 100 milliseconds, the eye starts to accelerate until it reaches the target velocity. And then when the target stops, the eye also decelerates and stops. So this is the kind of behavior that we use and I plan to use in order to study how reward impacts the eye movement system. Of course, maybe I'll go over this, although in this forum, I think we all understand that eye movements are really important and really excellent system for studying behavior, but still I'll mention the advantages that I see this and how it's important how it could be useful for studying a reward. So the eye movement system, of course, it's a simple behavior with only 6 muscles. Another advantage is the control we have over behavior. We can measure behavior very precisely and can easily manipulate the eye by moving the target. So it's easy to control. In addition, it's an excellent model system for trying to understand how different parts of the brain work, because it requires many different areas. So we can ask specific questions about not just what an area does generally, but specific questions, how an area is involved in a specific behavior. In addition, it's a smooth pursuit is a nice example for a sensory motor transformation. So we can study the whole system from the visual areas to the motor areas and study the full transformation. And we can go now and this is especially important when we try to understand what reward does, because we can now go into what we know these areas do in order to try ask how reward it changes the computations. So we were interested in studying a reward in the smooth pursuit eye movement system, and here you can see the very simple task that we developed when I was in the lab of Steve Lisberger. In this task, a monkey initially fixates on a target, then the target changes to color, either blue or red, and then after a random delay, the target starts to move. The monkey is required to accurately track it. And then in the end of the trial, if the target was blue, he gets a large reward, and if the target is red, he gets a small reward. And this is a very simple way, inspired by, of course, the work of Kohido Ikosaka and Reza, of course, a way that in the Saccade system, and we adapted this approach for the pursuit system. And this is the kind of behavior that we see. So this is an example for two trials. One in which the monkey is receiving a large reward in blue, and one that is receiving a small reward in the end in red. And what you can see here is that when the monkey is expecting a large reward, he accelerates his eyes faster and reaches the target velocity earlier. Whereas when he's expecting a small reward, we see a weaker acceleration. And because of that, he needs now to make these corrective saccades in order to reach the target. So we see this difference in behavior. The way we quantify this is look at the beginning of the behavior before the monkey makes these corrective saccades, and we simply average the eye velocity or look at the peak eye velocity at this time. And we can then show this, then we can compare the eye velocity for the large reward and the small reward. That's what you see here. Each dot here represents one session of behavior in one of our monkeys. The different colors are different directions of movement. And what you can see are very consistently across sessions, the eye moved faster for the large reward, when the monkey is expecting a large reward. And so we saw this in this monkey, and this initial studies we did in the US, then I opened lab in Israel, and we found this very robust behavior also in the monkeys in Israel. So this behavior apparently generalized between the US and the Middle East, which is nice. So this is the kind of behavior that we want to study. And as I said, that we're interested in understanding how the reward interacts with the smooth pursuit system. So when I opened my lab, one of our first project was going to look at how reward impacts the cerebellum processing. And at the time, there was some initial studies that told us that there might be some relation, a representation of reward in the cerebellum. So we used this task and studied and recorded activity in the cerebellar flocculus and the areas nearby. And we recorded the simple spike and complex spike. You can see a nice example for recording for my lab. And just to remind you, initially, in the cerebellum thought until recently is that these complex spike code the error, tells us when something went wrong. And initially, to our surprise, we found that in this task, in fact, what they code, they are modulated in correlation with the reward size. So here you can see the population response of the complex spike when the monkey, just after the monkey sees the cue that tells it what the amount of reward it will get at the end of the trial. And you can see here that when monkey is expecting a large reward, we see this increase in the complex spike, and in trials where it's expecting a small reward, we see a lower and perhaps a small decrease in the complex spike. So this was nice. This was, as I said initially, this was surprising. Another interesting fact is that when we looked at the output, the time of the reward delivery, we did not see a big difference between the large reward and the small reward. And this is at this point, the monkey is already expecting a reward. And this was similar to what how, for example, dopaminergic neurons code a reward. So at the time we did this study in my lab, at the same time there were other labs also studying reward, and there are many recent studies that showed that a reward is coded in the cerebellum and different parts of the different populations of the cerebellum, different species. Of course, there's a nice work. We were doing this the first in monkeys. Of course, there's a nice work of Mickey Goldberg that also showed similar results in other parts of the cerebellum. So these are this is what we initially found. But when I came before I did this study in my already in PhD, I already studied reward processing in the basal ganglia. And although this was new for the cerebellum, when you think about this is not extremely surprising when we think about processing in the brain. When you think about wherever you look in the brain, almost you can find a reward signal. So beyond that, this initial surprise, the question is how can you go forward? How can you understand deeper what the reward is doing in the cerebellum and how reward is processed across different areas in the brain? So there are several ways to go forward. The first one is I think is important, especially for people that come from the cerebellum that appreciate how important it is to understand what an area does. I think perhaps the cerebellum is one of the nicest areas where we have this tight link between neural activity and behavior. So maybe we can use this in order to understand not just find reward signals, but also deeper understand how they are related to what an area does, how they're related to computation and the transformation in this area. And initially, when we designed this test, that was the basic idea. We knew these cells are related to the computation of what we call retinal sleep. And we were interested in understanding how this retinal sleep is modulated by reward. So in this plot, you see what I mean by retinal sleep. So the x-axis is the time from motion onset, and the y-axis is the eye velocity. And the two colors show again the two conditions, large and small reward. And you can see here again this small difference between small but very consistent difference between the large and small reward and velocity. And the retinal sleep is simply the difference between the eye velocity and the target velocity. And we know that a complex spike in the cerebellum are modulated by this retinal sleep. And we're interested in looking whether the complex spike signal reward modulates this complex spike signal. But a bit to our disappointment, we didn't find much. So here you can see the response, the population response of the complex spike during motion onset in the preferred direction of these neurons. And you see there's not much modulation of the complex spike at this point. And we tried many other things. We also looked at simple spike, and we tried really hard to look for not just finding reward modulation, but trying to link it to what we know about the cerebellum. And we mostly failed in this. We mostly did not find any meaningful relation beyond finding the reward information. So this leaves us at this point a bit unsatisfied because we want to understand the system deeper. So the question, how can we go forward? So the first possibility, which I will present in the second part of the talk, is to use a comparative approach. So we're really focused on the cerebellum. We really like the cerebellum, but the brain is a bit beyond the cerebellum. And we can ask at the system level how different areas code a reward and movement. So that will be the second part of the talk. And in the third part, we still need, we'll go back to this focus on trying to understand the modulation of reward and what we know an area does. And we will ask how reward modulates the motor learning. So here's again the eye movement system. This is from the nice review of Rich Krauslitz, where we reviewed the relation between pursuit system and saccade system. And I'm looking now, here is a very highly simplified version of this plot in a box and arrow. So I'm sorry if I removed your favorite box or arrow. For simplicity, I did this. And as I said, we're interested now in understanding in a comparative approach where we will compare between the basal ganglia and cerebellum. And what you can see just by looking at the main anatomical pathway is that it seems that the cerebellum is closer to the generation of the eye movement. In addition, we know from stimulation studies that we can generate eye movement by stimulating the cerebellum by stimulating the cerebellum. However, stimulating the basal ganglia does not simply generate saccades. You need a more complicated context to show the effect of the stimulation. So, for example, during memory-guided saccades, we can see the effect of the stimulation, but not during visually driven saccades. So, based on anatomy and what we know about stimulation, what our initial thought was that in the basal ganglia that we know is so related to reward, we'll find these large reward signals. But perhaps in the cerebellum, we'll find signals that are more related to movement. So we could think of this as this hierarchical transformation of reward information into movement, and which reward is mostly perhaps represented in the basal ganglia, and perhaps in the cerebellum we'll see smaller signals related to reward, but larger signals related to movement. So that was our initial thought about the system. And in fact, there are very few studies that actually directly compared between the basal ganglia and cerebellum. So our goal was also to come up with a paradigm and an experimental setup that will allow us to compare these two areas. So what we did is record in the same monkeys in both the basal ganglia and the cerebellum. And again, we're interested in eye movement, so we focused on the eye movement areas of the basal ganglia and cerebellum. So in the basal ganglia, we recorded in the input structure that is related to eye movement, the caudate, and at the output structure related to eye movement, the substantia nigra pars reticulata. At the same time, we recorded also in the vermis of the cerebellum, and in the cerebellum, we were able to identify many neurons with complex spike, which tell us these are Purkinje cells. Often we recorded the other cells that did not have complex spike, and often had very different statistics in the firing rate. We think these neurons are not projecting out of the cerebellum, so we call these local neurons. So this is the basic recording setup. The task that we use is now very similar to the task that I showed before, but in this case, we're using probabilistic reward, not a reward size manipulations. So as before, the monkey initially is fixating on the dot, and the dot can change color, and then the monkey is required to track it accurately. And if the dot was blue, he gets 75% of the trials, he gets a reward, and 25% of the trials it's omitted. And if the dot is red, only 25% of the trial, the monkey receives a reward. And we recorded in this task, we recorded both saccades and pursuit, both saccade and pursuit, to have a more comprehensive comparison between the two areas. And you can see here the behavior. So during pursuit, as I said before, the monkey smoothly tracks the target. During saccade, after a short the target jumps, and after a short latency, the monkey makes a saccade toward the target. So overall, what we have here in this experimental design is recording basal ganglia and the cerebellum in the same lab, in the same monkeys. Often we also record simultaneously in the two areas, using the same kind of task, pursuit and saccades. And of course, in the same, we're interested in the same motor system, the eye movement. And this similarity between all these parameters will allow us to directly compare between the basal ganglia and the cerebellum. So before I go into the details of the comparison, I want to tell you how I did this comparison. So the way in which we compare the modulation and the activity in these two areas is using effect size. We used a well-known measure of effect size called omega square. Intuitively, what this measure does is quantify the part of the variability in the activity that is related to the parameters of the task. So for example, you see the square perhaps representing all the variability of a neuron. A part of this variability can be explained by the parameter of the task. For example, could be a manipulation of the reward probability or different direction of movement. So part of this variability we could explain. This is, for example, be the yellow or the pink part. But other parts of variability that are not related to the parameters that we manipulate, and this is this part. And what the omega squared does is calculate the ratio between what we could explain and what we could explain plus all the noise. So this is essentially a kind of a signal-to-noise error for a single neuron. So, for example, if all the activity is related to one of the parameters of the movement, let's say this could be, for example, a cell responding really, really strongly to the direction of movement, then this measure is close to 1. When only tiny parts are related to the parameters, then this measure goes close to 0. So the measure of effect size. And specifically, omega square has the advantage that it's unbiased. It deals with opposite effects. If the cell increases or decreases, in both cases, the omega square, the parts of the activity that are related to the task will be mapped into positive omega squares. In addition, we verified this measure is relevant for neural data by showing that it correlates with the performance of simple decoding of the parameters of the task. So now we have a measure now we have a way to compare between the different populations. And here is an example for the calculation of the omega square. So here is an example for a neuron from the output of the basal ganglia from the substantia nigra pars reticulata. On the left here, you can see the average response, the PSTH of this cell for the condition we're expecting with a large probability and small probability, and blue and red. And you can see this cell responds very strongly at the time, just after the monkey sees the cue that tells him about the reward probability. And here you can see in the middle, you can see the raster of this neuron. And you can see this response is very consistent across many trials. When the cell increases, it increases very consistently. And when it decreases, we see this pause in most of the trials. So then we use this, we can bin the activity in different times and calculate in each bin the omega square. And as expected from what we see activity, the omega square is before the omega square related to the reward probability. It's before the Q appears as close to 0. Then it increases when we see this large response, and then it decreases back to 0. So this is one measure of effect size. This is the effect size in time. We also measure the overall effect size. In this case, it's around 0.5. Okay, so now we have a measure that you understand, which we will use in order to compare between the different populations. So initially, we compare the activity, the effect size in the different populations at the time of the cue. This is just when time 0 here is just when the monkey sees the cue that will tell it what will be the probability of getting a reward. And the y-axis here is the average effect size across the different populations, and different colors are different populations. And what you see very clearly here is that the effect size related to the probability of reward was very large, or largest in the substantia nigra reticulata, the output of the basal ganglia. Then it was intermediate in the cerebellum and the different population. The cerebellum was smallest in the input structure of the basal ganglia.
Maddie, may I ask you a question?
Yes. Yeah, of course.
Purkinje cells are simple spikes here you're referring to?
Yeah, I'm here referring only to simple spike. We did this analysis also on complex spike, and the responses are much, much, much smaller, and everywhere, all the time. If you think of the effect size as a measure of the signal to noise, so the measure of signal to noise of the simple spike was an order of magnitude larger than the complex spike. I'm not showing them here, and we have in the bioarchive, we have a paper about this, and you can look there and you see the analysis for the complex spike. Okay, so this is the effect size of the 4 different populations. Here you can see the single neurons, and you can see that in the output of the basal ganglia, we see many neurons with a very, very large size related to the reward probability. As I said, this here below, this just summarize this. This just summarize this finding. We find that the output of the basal ganglia was largest than cerebellum, both populations, cerebellum intermediate, and the basal ganglia input was smaller. And they want to emphasize, although small in all populations, this was larger than 0. So this is confirming what we know both about the basal ganglia and cerebellum. There is a modulation that are related to the reward in these areas. And again, we thought initially, the way we thought about it initially, perhaps we'll find one thing in the basal ganglia and the other in the cerebellum. And this was not the case. We find this more complicated picture about recoding of the reward probability. In the next step, we went into the movement itself and calculated the effect size during the movement. And during the movement, now we have two parameters. One is the reward probability, and the other one is the direction of the movement. And here's an example for a neuron. This is a neuron that in the cerebellum that did not have a complex spike and very different kind of statistics of firing that you're used to see in the simple spikes. What you see here, x-axis is the time, the different columns, different direction of movement, and the different colors show the different reward probability. And what you can see that this neuron responds very, very strongly when the eye moves to the right towards the 0. But if, for example, did not respond at all when the eye moved to the left. So it's strongly modulated by the direction of movement. But when we look at the reward, we don't see much modulation related to the reward, since the responses are quite similar for given a direction, the different reward probabilities. And this is indeed reflected in the effect size. So the effect size can allow us to partition the different sources, the different parts of variability, and plot each of them separately. So you can see here the large increase in the effect size related to direction. You can think of this as the tuning of the neuron, and not much modulation in this neuron related to a reward. So then we wanted to compare between the effect size and the different population during the movement. And this is what we found. X-axis here is again time. The vertical axis is the effect size of the direction of movement. Different colors correspond to the different populations. And what you see here, I would say to our surprise, that we found that the effect size related to the movement, to the direction, was largest in the basal ganglia, output. Again, it was intermediate in the cerebellum and smallest in the basal ganglia input. So again, initially we thought perhaps the cerebellum will be more tightly related to the sensory motor parameters of the movement, but in fact, what we found is the output of the basal ganglia was larger. So again, we found this order of the basal ganglia output being the largest, the cerebellum being intermediate, and the basal ganglia being input smallest. In this case, all the populations were much larger than 0. In this case, it's not very surprising, because this is how we chose the areas to record. We looked for areas that are related to eye movements. So this was not surprising. It was again, the surprise for us was that we found this very large response in the basal ganglia output. Just to say, when we look at the reward coding during movement, we actually found quite very small modulations related to the reward during the movement itself. One control that is important, because I'm often asked about this is not simply a result of the firing rate of the neurons. So different populations have very different baseline firing rate, but we did not find any clear correlation between the firing rate of the neuron and the direction effect size. As I said, this is a measure of signal to noise. It's not a measure that is directly related to the firing rate, and as a result, we do not see any clear correlation between the two.
May I interrupt
yes.
for a question?
Yes.
Did you check to see if these Purkinje cells are saccade-related cells, or any kind of Purkinje cell that you could record from the cerevermis you have included in your analysis?
Yeah, so we did that. So what is nice in this that measuring the effect size, we can actually compare exactly these kind of questions. As does a cell respond to a saccade or to pursuit? I have it here. Maybe I'll show it in the end. But the short answer is that these cells respond to both saccades and pursuit. So overall, the correlation between the effect size for pursuit and saccade itself was correlated. So cells that tended to respond stronger to pursuit also tended to respond stronger to saccades. Okay, so we were talking about this correlation. I said that we can compare. We can use this to compare between the responses across different tasks. These measures. So how can we think about this? What's happening here? We know that both in the basal ganglia and in the cerebellum, there's huge convergence from input to output. In the basal ganglia, there are many neurons in the striatum, but much fewer in the output, in the substantia nigra reticulata. In the cerebellum, of course, there's this famous convergence from many billions of granule cells to much fewer Purkinje cells. And anatomically, we know there's this convergence. The question is, what do we know functionally about this convergence? And I will maybe a way to think about this is we have here an output neuron, and this neuron gets many inputs from many neurons. We can think of this as each eye represents a single neuron, and the activity of each neuron is part of it is related to the signal, which in our case is the coding of the specific parameter, task-related parameter, reward or direction, and part of it is not related to this. This is what we call noise. And during this convergence, there are two things that could happen. One possibility is that the signal will be sharpened. This could happen if the signal itself is correlated across many neurons. Many neurons are coding the same parameter, but the noise itself is uncorrelated. And as a result, we'll get a averaging out of the noise. This is what I call signal sharpening. But this is not. It's not necessary that we'll have a signal sharpening. We can also have no sharpening or even cancellation of signal. This will happen if the signal is uncorrelated. For example, if cells with opponent responses converge on the same neuron, or if noise is correlated. In that case, we won't get this sharpening. So our results, so what we see in the basal ganglia, we see this very strong sharpening of signal, whereas in the cerebellum, we see that these local elements and the output, the size of the signal is very similar. So what we're suggesting is perhaps we don't need to think about the basal ganglia cerebellum as having different roles in movement or reward, but the difference is in the way in which the signal is processed. So perhaps in what we see in the basal ganglia, we see this sharpening is because what is happening is that many neurons with the same kind of property response in the input on converging on the same output neuron. So this is demonstrated here. So these the circles on the top represent the input neurons, and the circles on the bottom represent output neurons. And then the input neurons, there's the gray part represents what we think about noise, and the black part is one signal, and the white part is another signal, perhaps with opponent properties. Then many black neurons are projected here on a single output neuron. And as a result, the signal that we see in the output is much larger than the noise. Whereas perhaps what's happening in the cerebellum, but we don't see this sharpening signal. Perhaps this is happening because there's different signals with opponent properties are converging on the same neuron. So this is represented here, where on this output neuron we have many neurons that prefer this, let's say, black signal, but from the same neurons. But the same neuron also get this neurons with the second signal, as you can see here. And as a result, because we have opponent signals, this signal will be averaged out in the signal itself might be also averaged out. And as a result, we might not see an increase in the signal to noise. And one question, I think, critical question, what is it good for? So sharpening signal is, of course, good for extracting signal. We do it, we know from many algorithms need this. This is used for many algorithms, for example, PCA. This is, in fact, what the motor system does in order to drive behavior. We know that closer we are to the behavior, the signal to noise of the neurons is higher. So there are many roles in which we need a sharpening signal. But when do we need this convergence of an opponent signal? This could actually be very useful for when you think about it, for learning, because the connections are already there. And now if we want to choose them, we can easily, a neuron can, based on learning rules, change these signals. So this perhaps could be good for learning new behavior, or rapid learning of behavior, I would say. So this is this part concludes the part where I wanted to compare between the basal ganglia and cerebellum. So the first we talked about reward signal in the complex spike and the cerebellum, and then I showed this work in which we studied convergence. Do I have like another 10 minutes for third part? Yeah, okay. Okay, but still, I think again, I want to emphasize this because this is I believe in this deeply, that we should not just look for reward signal. We need to try to understand how these reward signals are related to what we know areas do. So in that direction, one thing we know, so we still do not know how reward interacts with what we think of as cerebellar computation. And one thing we know that cerebellum does is learning, is motor learning. So I think the next part, I'll show you a behavioral study in which we try to ask how reward impacts what we think is cerebellar dependent behavior. So for this task, again, it's only behavior from now on, no neurons. But what we did for this task, what we did is try to ask how reward impacts learning in the pursuit system. So the basic behavior task that was used for learning in the pursuit system is, you can see here. In this task, the monkey initially fixates on the dot, then the target starts to move, and then without any other cue, the target changes direction. So we can define the base direction, the direction in which the monkey initially moves, as in this case, the horizontal direction, and we can define the learning direction as the direction orthogonal to this movement. That's what you see here. The learning at which the direction in which the target will change direction. So in this task was developed by Javier Medina, Megan Carey, and Lisberger in the lab of Steve Lisberger, already 17 years ago. And what you typically see in this task is this kind of behavior. So the x-axis here is time from the motion onset. The y-axis is the velocity in the learn direction. The arrow here represents the time when the target changes direction. And what we see after several learning trials is that the monkey starts to move, and also humans, towards the direction in which it will change. And this is what you see here. So this part, what I'm marking in gray, this is the behavior, this is the predictive behavior. This is the behavior the monkey does before the target changes direction. After the target changed direction, you can see the behavior here. There's this reaction to the change in direction. The monkey accelerates towards and reaches close to the target velocity. So to quantify this learning, what we do is average the behavior at this window just before the target changes direction. And here you can see a typical learning curve. So beginning of we see this, typically what we see is that there's this fast learning that takes several trials, around 10 trials, that the monkey learns to move towards the direction in the learned direction. And then there's another slower learning across many trials. In this case, we stopped here at 100 trials, but it also increases slightly beyond 100 trials. So this is the basic task, and this is the basic kind of learning. And we know that in this task, there were very nice correlates of the learning in the cerebellum. And we wanted to ask whether we can, how reward modulates the learning in this task. So what we did here is add the reward manipulation to this task. So initially, the monkey is fixating on the target. And then when the target starts to move, it changes color, either to blue and red. And then in these trials where the target changed to blue, the target it changes direction later upwards. And in these trials, the monkey gets a large reward. In other trials, the target will move downwards, is red, and it will move downwards, and then the monkey will get a small reward. And well, the kind of task that we did in order to probe the impact of learning is use a random sequence. So we randomly interleave trials with these when the target moved up and down, and ask how the eye is impacted in this random sequence. And here you can see the basic learning that we found. So we had two kinds of blocks. One block in which the large reward moved up, and the small reward target moved down, and another block where the large reward moved down and the small reward moved up. And here you can see the behavior, the x-axis again, time from motion onset. This is all the predictive behavior. This is not the behavior after the target change direction. And the y-axis, you can see the velocity in the learned direction. And what you see here, that in blocks, when the large reward moved up, the monkey moved up in these trials, but he also moved upwards when he was in the small reward trials. So this is important. These are trials when the small reward target initially moved downwards, but still at the initiation of the movement, the monkey moved upwards. And we saw the opposite picture when the large reward moved downwards. You can see this here. And to really show that so there are two associations here. There's one association between the color and the direction of movement, and the association between the color and the reward. to really show that learning is impacted by the reward. We had probe trials when the monkey, the target never changed direction and did not change color, and it always moved straight. And we had these probe trials in blocks where the large reward moved up, or in blocks where the large moved down. And what we see very clearly that in blocks where the target moved upwards, the probe trials, the monkey moved up, and when the trials where the probe trial moves down, the monkey moved. When the large reward moved down, the probe trial the monkey moved downwards. So reward clearly impacts the learning in this task. One thing that we were interested in studying whether the timing of the reward information is important. So what we did in this task is signal at different times in the task when the size of the upcoming reward. So up to now, I showed you this condition, what I call onset, when the target started to move, it changed color, but we also had condition that ahead of time, the monkey knew what the reward will be and the direction of movement. This is what we call fixation. We had also condition with that just when the target changed direction, that's when the target changed direction, it changed also color. And we also had a condition with no cue that when the monkey moved upwards in the end of the trial, he got a large reward, or downwards, for example, he got a small reward. And what we found, again, to our surprise, is that we found clear learning only in the condition when a reward. We found the impact of the reward only in the condition when the color changed at motion onset. And you can see this here in purple. These are also the plots that I showed before. Only in this purple condition, we see differences in conditions when the large reward moved upwards or when the large reward moved downwards. And so now we have a task that we know that we think it depends on the cerebellum. So in this task, as I said before, there are very strong relations between the learning, that very strong correlates of the learning in the activity of the simple spike and the complex spike. In this task, it seems at least that the learning follows the rule of the classical Mar-Ito models, in which a complex spike in one trial impacts the simple spike in the next trial. And what we want to do now is go and look whether this learning that we think depends on the complex spike and is modulated by reward will be modulated, will indeed be modulated, the complex will indeed be modulated by reward. And perhaps it will be a nice way to show that how reward impacts the computation of the cerebellum. So we expect that we have this signal related to the complex spike when the monkey sees the cue that tells about the upcoming reward, and we have this signal in the complex spike related to the movement. Perhaps when these two signals come in close proximity, we will see some summation of this signal that will potentiate the learning. So to summarize everything, I talked about reward in the cerebellum. Then I talked about our recent study in which we compared between the basal ganglia and cerebellum and coding of reward and movement. And in the end, I showed a new task that we developed in order in the future to be able to study how reward impacts processing in the cerebellum. And the questions, of course, the people that contribute to this. So these are the people in the lab that actually did the work. Adid and Merav did the recording and the initial recording of the cerebellum, the complex spike. Then Noga and Gil did the recording of the simultaneous recording of the basal ganglia and the cerebellum. Judith and Yirat are working now on pursuit learning and the impact of reward on pursuit learning.
Maddie, thank you so much. That was just magnificent. So many beautiful, thought-provoking experiments. You're certainly on your way to try to make sense of how reward information affects behavior, particularly learning. Let me start the questions by going back to the basal ganglia cerebellar part of the presentation and ask you about whether there is. You didn't talk so much about your presentation focused on the simple spikes, and so I just want you to give you a chance to sort of connect the information that you showed with regard to complex spikes and the variance measure that you were describing for the simple spikes. And can you give us a little bit more information, clarify that?
Yeah, so I don't think I have the figure here. But we did this measure of effect size in complex spike, and as I said, it was tiny. If you think about it, and whoever measured complex spike knows that although you can really, if you really try hard, you can extract information from the complex spike, but it's extremely noisy at the single cell level. So this measure of signal to noise in complex spike was very small. We did find some responses related to the Q. We also found, of course, responses of complex spike related to the movement itself. Again, we went to an area we expected these kind of responses. But I think the main issue was much smaller than the responses of the simple spikes.
Thank you so much.
As I said,
thank you so much. Are there any other questions? Let's hear other questions. Violetta, go ahead.
Yes, indeed. Thank you for the nice talk. I was curious, the local neurons that you mentioned, were they also from the deep cerebellar nuclei? Or
no, they're not. So yeah, maybe I'll go. I have it now.
Since there are these new findings that they are connected to many of the basal ganglia, I was thinking that maybe they're a good candidate to look for these questions.
Yeah, so they're not deep cerebellar nuclei. So we didn't try to classify them. I think Reza, you have recently, I think, recorded with probes in the cerebellum. I know there are other labs that are doing this. We were recording with single electrodes, and with single electrodes, it's really hard to try to classify which cells these exactly are. We have some hints. So, for example, here you can see the firing rate, and in the direction effect size is a function of the firing rate. And these green cells are what we call local neurons. We have some neurons with very low firing rate, base firing rate. Maybe these were called in the past MOSI fibers. I'm not sure, I don't know, but it's possible. We had other neurons with higher firing rate, and we have some more regular neurons. Again, we did not try to classify them, but it seems it's a combination of different types of neurons. Clearly not deep cerebellar nuclear neurons. We were recording in the cerebellar cortex.
Thank you. Amir has a question.
Hi, thank you for your beautiful presentation. I just have a question about a slide about the convergence of signals. You make a comparison between basal ganglia and cerebellum, and I was wondering that in the cerebellum part you make a figure that shows that cerebellum has a machinery prepared for learning role. And I think maybe, I don't know, if we, record the data or measure the effect size formula that you computed and to compute this simple spikes from the nucleus rather than the input from the Purkinje cells, maybe this role has a change, I see that there is I think there is lots of MOSI fibers that are parallelly they are inputting to the Purkinje cells, and maybe if we measure this effect size from outputs of the Purkinje cells, we can see some activities like PCA like the basal ganglia part you mentioned there.
Yeah, of course, that's possible. I don't know. It's possible. It will tell us. I think it's very important to look at different stages across the different areas and ask how the effect size changes. I think that will tell us about the rules in which signal converges at different states. I don't know what happens in the deep cerebellar nuclei in this task. I would be very interested in it.
Yeah, thank you.
Abby, please go ahead.
Thank you so much for that beautiful talk. I was curious about the part in the recordings in the substantia nigra pars reticulata, where you showed those beautiful examples of where the SNR cell was firing much more in terms of rate to the high probability reward relative to the low probability reward, if I remember that right. So my question has to do with whether the vigor of those subsequent movements differs, and if it's higher in the high sort of probability reward situation, are those rates that you see in SNR somewhat paradoxical given the general model of disinhibition of targets?
Yeah, so yeah, that's an excellent question. I have a lot to say about this. So in terms of the, we did not see any consistent increases or decreases across different probabilities. We saw quite a miss in that. But I want to emphasize another point about the kind of miss that we found in this SNPR, the output of the basic ganglia. So the classical idea is that, of course, this idea from the work of the Kosaka and Wirtz, where the basal ganglia disinhibits, the SNPR disinhibits the superior colliculus and allows a movement, right? And we find some neurons that respond like that. We find some neurons that have during the movement this inhibition of this clear inhibition. But we found also cells that were complex in ways that were much beyond what we expect. So here's an example for a neuron. This is the response of this neuron to 8 different directions of movement during pursuit. And you can see this is the response. And just looking at this looks like noise. But in fact, when you look at the trial by trial basis, you see that this noise is what looks like noise is actually very consistent response of the cell. So, and I didn't talk about this at all, but we compared this complexity to other areas, and in the cerebellum, we have also neurons in the cortex that we recorded, and the output of the basal ganglia was very complex, more complex than any other structure that we recorded, and huge responses. I think this is very interesting and important.
Hi. I have a question. Thank you very much for your beautiful presentation and beautiful data. And I have a question about the last data you showed, the learning in the cerebellum and learning in the pursuit, the direction change. And you showed in this slide you showed the even in the downward direction change, you showed the monkey learn the upward prediction, right? It is surprising because I expect this learning should be driven by the complex spike and driven by the movement error. So I expect the movement error should be downward, but this learning is upward. So it's a kind of paradoxical. So I expect some interaction between the upward learning and downward learning. So do you have any idea why this kind of the paradoxical learning?
Yeah, thank you for asking the question. Excellent question. In fact, I wrote a grant about doing exactly that. We did not have the data itself. So if you want to know the answer and you happen to review the grant, then you can help me and I'll do it. but that's exactly the point. We have this response. We have these responses. We think this is driven by complex spike. We know that the reward impacts this complex spike. Perhaps this interaction between reward and complex spike is the reason that we see this learning upwards, although this we call paradoxical learning. So imagine at the extreme conditions that the reward modulation are so large that only in trials when the target moves upwards, it gets there's a complex spike, but not when it moves downward. And that will lead to movement, that will lead to learning that will always be only upwards. So that's the prediction, and thank you for asking.
Okay, thank you so much.
Thank you, Shogo. And Leah has a question.
Hey.
Hi, thank you for that excellent talk. So I have a question following up on Abby's question, and you may have already answered it in your response, but it was with regards to vigor. So in your experiment, you have, in the first set of experiments you described, there was a saccade task where the monkeys were saccading to either low or high probability of reward. And I was curious, did you see any difference in the reaction time or the vigor of the saccade?
This is excellent. Again, excellent questions. That there was a fundamental difference between the task. I did not talk about it in terms of vigor. Let's see if I can have it here. So one thing we notice is that when we use large and small reward, we see very, very clear differences between different conditions. Okay, we see that monkeys really, and you also see this in the behavior outside of actually doing the behavior. They really like the large reward. They really hate the small reward. And in the behavior, you see this much larger, and we see this larger velocity for the large reward. But when we use the probabilistic task, the picture was more complicated. And here is an example. Here you see the behavior of one monkey. We in fact measured 5 different probabilities in this monkey. You can see here the pursuit initiation. And often what we saw is that modulation by the probability was quite small in some probabilities. So for example, in this behavior, the probabilities 1 until 0.25 were quite similar in terms of the gain of the pursuit, perhaps slightly larger for the some conditions, but there was a big difference between that and the 0 condition. Okay, and this is quite, and this is in one monkey. In the other monkey, we found a slightly different picture, but the monkey don't seem to code in their vigor of the pursuit or in the saccade linearly the probabilities. We see these differences in sometimes. In this monkey, you can see here, this is the in blue is the pursuit velocity. So 0 is the smallest, and then it increases for the other probabilities. And in black is the saccade latency. So it's long for the 0, but larger for the others. It's longer for the 0, but smaller, shorter for the other probabilities. And I want to emphasize, this is not that the monkey don't understand the task. We have also a target where the monkey selects between the two probabilities, and you can see here the confusion matrix. In all conditions, when we, for example, compare 25 to 75% of receiving a reward, when they need to choose between these two targets, in almost 100%, they choose the 75%. In all different conditions, in around the worst case was 80%, the monkey always chose always preferred the larger reward probability.
Okay, so choice is modulated by probability, but you're saying in your results, vigor is really just binary, reward or no reward. And this is
I'm saying in this monkey, other monkey, it's slightly different. It's very sensitive, I would say.
Okay. Well, yeah, thank you. That's really interesting.
Yeah.
So Nazanin has a question.
Yeah, thank you for the presentation. I just wanted to quickly check if I'm getting the big picture right. We used to, as long as I know, we used to believe that basal ganglia are considered as the reward center of the motor system, and cerebellum as the part that just adjusts the sensory motor parameters or does some error-based motor adaptation. So what we did here is that we kind of challenged this view and said that it's not that straightforward. This whole separation of roles is not correct, because we saw that, okay, the reward size, for example, affects the complex spike rate and things like that. Am I getting it correctly?
Yeah, and I think there's a point that I do want to say here. I think that looking at existence of signal is not enough. And you'll find signals distributed across many different circuits. I think what we need to think about is rules of transformation, and that's what that's what I try to convey in this picture. That we see reward and movement in both areas, but it seems that the way in which their process is different. I think perhaps that will be the way to look now across different areas and then try to understand how information is transformed. That's one thing. And the second point is that we really need to try hard to link these modulations to what we know already about areas and try to link it to the computation, because otherwise it's just hunting signals. So that's my comment about that.
Yeah, I get it. Thank you. Thanks for the presentation.
Maddie, thank you so much. That was really thought-provoking, and we're so blessed that you're doing these wonderful experiments. Okay, everybody, have a wonderful Tuesday.
Thank you.